Think for a moment about what keeps you healthy. You probably consider what you eat, maybe how much you exercise. But what about the things you can't see? The microscopic hitchhikers that could be lurking in your glass of water or on your dinner plate. It's a sobering thought, but understanding these invisible enemies is the first step to staying safe.
That's right. We tend to focus on the big, obvious health risks, but the reality is that a vast number of illnesses are caused by these tiny bacteria and viruses that we encounter in our everyday lives through water and food. Being informed about these threats isn't about becoming paranoid. It's about having the knowledge to make smart choices and support effective public health measures.
And that's precisely what we're diving into today. You've compiled a really comprehensive collection of information all about waterborne and foodborne bacterial and viral diseases. Our goal here is to navigate through that material together, pulling out the most important insights, those aha moments that help us truly understand these issues without getting bogged down in overly technical details.
Exactly. You've laid the groundwork by gathering this information. Now we'll work together to connect the dots, highlight what's most crucial for you to know, and hopefully even reveal a few surprising aspects of this microscopic world.
Okay, let's start with something fundamental. Water. It seems so clean and essential, but as our sources point out, it can also unknowingly transport some unwelcome microbial passengers.
Absolutely. Water, whether it's what we drink from the tap or what we swim in at the lake, can serve as a vehicle for a wide variety of disease-causing microorganisms. This is why the work of water quality professionals, the engineers and microbiologists, is so vital. Think of them as the guardians of our water supply, constantly monitoring and ensuring its safety.
And when we consider drinking water in developed countries, it's easy to take its safety for granted. Our sources detail the strict quality controls, the filtration processes, and then this key step, chlorination. Can you explain why adding chlorine is such a game changer for water safety?
Sure. Chlorination, typically using chlorine gas, is a cornerstone of modern water treatment. It works in two important ways. First, it actively kills any harmful microorganisms present in the water. Second, it leaves a small residual amount of chlorine in the treated water as it travels through the pipes to our homes, providing ongoing protection against recontamination.
Ah,
Water that meets these rigid standards is what we call potable, basically, safe to drink.
it's good to know there's that double layer of defense. But the information also notes that even with these measures, outbreaks can still occur, and Legionella is specifically mentioned. What makes this bacterium such a persistent challenge in water systems?
That's a critical point. While our drinking water is generally very safe, no system is perfect. Legionella is a type of bacteria that, well, it loves warm water and has the ability to form biofilms. These are like slimy cities of microorganisms that can stick to surfaces within water pipes and tanks, making them harder to eliminate with standard disinfection.
Biofilms, right.
So even in well-treated systems, if conditions are right in places like hot water heaters or complex plumbing networks, Legionella can multiply and then get released into the air in tiny droplets, which can cause infection if inhaled.
So it's more about breathing in contaminated mist than actually drinking the water when it comes to Legionella. Now let's shift to recreational waters, lakes, rivers, and pools. Surprisingly, our sources indicate that these actually cause more outbreaks than drinking water. And they highlight that parasites are more often the cause in these cases. Why is that?
That might seem counterintuitive initially, but think about the nature of these water sources. Recreational waters are often open to the environment and can be contaminated by various sources, including both human and animal waste. While public pools in the U.S. have regulations, these primarily focus on chemical disinfection levels. Natural bodies of water like lakes and ponds have much less oversight. And yes, we do see a higher frequency of illnesses caused by eukaryotic parasites like Cryptosporidium or Giardia in these settings. These parasites often form cysts that are more resistant to standard chlorine disinfection compared to many bacteria.
And the information also mentions a significant number of gastrointestinal illnesses linked to recreational water where the exact cause isn't identified. It's like we know the water made us sick, but we don't know which bug to blame.
That's a real challenge for public health officials. Pinpointing the specific pathogen in every case can be difficult, especially if people don't seek medical care or if specific tests aren't conducted. It underscores the complex mix of potential contaminants that can be present in recreational waters.
Now, how do we actually determine if water is safe? Our sources detail water quality testing and this idea of indicator organisms. E. coli gets a lot of attention. Why is this particular bacterium considered a warning sign in water?
Well, think of E. coli, specifically fecal coliform, E. coli, as a kind of sentinel organism. Its presence in water strongly suggests recent contamination with fecal matter.
Okay.
Since many waterborne diseases are transmitted through feces, finding E. coli is like finding a clue that other potentially harmful microorganisms might also be present. It's a good indicator that the water might not be safe for consumption or recreation.
That makes sense. And the sources describe this membrane filter procedure using something called eosin methylene blue, or EMB medium. That sounds quite technical. What's the basic principle behind this?
Imagine a very fine sieve that traps any bacteria in a water sample. That's essentially what the membrane filter does. This filter is then placed on the EMB medium, which is a special type of growth jelly for bacteria.
Mm.
This jelly is selective, meaning it encourages the growth of certain bacteria like coliforms, while inhibiting others. It's also differential, meaning it helps to visually distinguish between different types of coliforms based on how they grow and change the color of the jelly. So if E. coli or other coliforms are in the water, they'll form distinct, visible colonies on this EMB plate, allowing scientists to detect and count them.
So these little bacterial colonies act like visual alarms, and there are also these defined substrate tests for specifically identifying E. coli. How do those work in a simpler way?
Think of these tests as offering specific foods that only the target organism, E. coli in the case, can eat. When E. coli consumes these unique food sources, it produces the detectable signal, often a fluorescent glow or a color change. This allows for a quicker and more precise way to identify E. coli compared to just observing how colonies look on a general growth medium.
The sources also briefly mention ATP detection assays using luciferase. That sounds almost magical. How does that help in assessing water quality?
It does sound a bit like science fiction. ATP, or adenosine triphosphate, is the energy that powers all living cells. The luciferase enzyme is the same thing that makes fireflies light up. When luciferase encounters ATP, it causes a reaction that produces light. So by measuring the amount of light produced when luciferase is added to a water sample, we can get a rapid idea of the total amount of living microbial material present. While it doesn't tell us exactly what kinds of microbes are there, it gives a quick overall picture of the microbial load. Think of it like a general indicator of biological activity.
That's fascinating. So, in regulated water systems, what are the expected results from these tests? What makes the authorities take notice?
In properly functioning regulated systems, the expectation is that tests for total coliforms, and especially E. coli, should be negative. That's confirmation that the treatment processes are working effectively and the water is safe. If either total coliforms or E. coli are detected, it signals a potential issue with the purification or distribution of the water, and that's when public health agencies step in. The EPA sets the safety standards, and water utilities are required to report any positive results and inform the public if there's a health risk.
That makes perfect sense. Now, the last point under water as a disease vehicle is quite intriguing and a bit concerning. Reverse zoonosis in Antarctica. Can you explain what that is and why it's a worry in such a remote place?
Reverse zoonosis is essentially when humans pass diseases to animals, the opposite of what we usually think of with zoonotic diseases where animals transmit to us. As human activity expands, even to isolated environments like Antarctica through research and tourism, there's a risk that we could introduce our own microbes to wildlife that haven't evolved any defenses against them.
Wow.
The increasing human presence in Antarctica raises concerns about potentially introducing novel pathogens to these unique animal populations. The discovery of human-derived enteric pathogens, including antibiotic-resistant Salmonella and Campylobacter in Antarctic marine birds, is a stark warning of this risk. It really emphasizes the need for very strict biosecurity measures to protect these fragile ecosystems from unintended consequences of our presence.
That's a really unsettling thought that even our seemingly benign activities could have negative impacts on these remote animal populations. Okay, let's now move on to the actual diseases transmitted through water. Our sources explain that these can occur from either drinking contaminated water or inhaling those contaminated aerosols, like with Legionella. And it's noted that this is a constant risk in areas where sanitation is poor.
Exactly. In regions lacking proper sanitation and access to safe drinking water, the threat of waterborne diseases is significantly elevated. These illnesses can have devastating effects, particularly on vulnerable individuals like young children and the elderly.
Let's start by diving into Vibrio cholerae and the disease it causes. Cholera. It's described as a severe diarrheal illness. What are the key things we should understand about this bacterium and how it makes people so sick?
Vibrio cholerae is a gram-negative bacterium that thrives in aquatic environments. People typically get infected by swallowing contaminated water or food, and the information specifically mentions raw or undercooked shellfish as a potential source. Interestingly, it usually takes a relatively large number of these bacteria to cause illness because our stomach acid is quite effective at killing them off.
Hmm.
However, if someone has reduced stomach acid, or if the bacteria are consumed with food that can neutralize the acid, a much smaller dose can lead to infection. Once V. coli reaches the small intestine, it attaches to the intestinal lining and releases a powerful toxin called cholera toxin. This toxin disrupts the normal flow of salts and water in the intestines, leading to the characteristic severe, watery diarrhea and rapid dehydration that defines cholera. If not treated promptly, this severe dehydration can quickly become life-threatening.
That rapid loss of fluid sounds incredibly dangerous. What's the primary approach to treating someone with cholera?
The most critical aspect of treatment is immediate and aggressive rehydration. This is done using oral rehydration solutions that contain a balanced mix of salts and sugars, or, in very severe cases, through intravenous fluids. Antibiotics, such as doxycycline or azithromycin, can also be used to help shorten the duration of the illness and reduce the amount of bacteria shed by the infected person.
And what are the key strategies for preventing cholera from spreading?
Prevention is a multi-pronged approach that relies heavily on public health infrastructure. Ensuring proper sewage treatment and providing access to safe drinking water are absolutely essential. On an individual level, practicing good hygiene, including frequent hand washing with soap and safe water, and avoiding untreated water and raw or undercooked seafood are crucial. In areas where cholera is common, vaccination is also an important preventive measure. Diagnosing cholera typically involves isolating and identifying Vibrio cholerae from a patient's stool sample in a laboratory.
Okay, a really serious disease with potentially devastating consequences. Let's move on to another bacterial culprit we discussed earlier, Legionella pneumophila, and the illnesses it causes, Legionnaire's disease, and the milder Pontiac fever. The way this bacterium is transmitted is quite different, isn't it?
Yes, with Legionella, the key is inhalation, not ingestion. Legionella pneumophila is another gram-negative bacterium that naturally occurs in freshwater environments. The problem arises when it colonizes human-made water systems, like the cooling towers of air conditioning systems, evaporative condensers, and even our home hot water systems, where it can form those protective biofilms we talked about earlier.
Right, the biofilms again.
The bacteria then get dispersed into the air in very fine water droplets, creating aerosols. If someone inhales these contaminated aerosols, the Legionella can reach their lungs and cause Legionnaire's disease, which is a type of pneumonia, or the milder flu-like illness known as Pontiac fever. It's important to remember that Legionella is not spread from person to person.
So it's not contagious in the traditional sense. What happens in the body after someone inhales these tiny Legionella-containing droplets?
The Legionella bacteria are inhaled into the lungs, where they can infect certain types of lung cells. They are also taken up by immune cells called macrophages. However, instead of being destroyed by these immune cells, Legionella has the ability to survive and multiply inside them, leading to inflammation and the development of pneumonia. The severity of Legionnaire's disease can vary greatly, ranging from a mild cough and fever to severe pneumonia that requires hospitalization, particularly in people who are older or have weakened immune systems.
How is Legionnaire's disease diagnosed and treated?
Diagnosis usually involves culturing Legionella bacteria from respiratory samples, such as sputum, or detecting Legionella antigens in the patient's urine. Antibody tests can also be used, but these often take some time to show a response. Treatment typically involves antibiotics that are effective at penetrating lung tissue and the macrophages where the bacteria reside, such as azithromycin, levofloxacin, or doxycycline.
And what measures can be taken to control Legionella in water systems and prevent future outbreaks?
Control strategies focus on minimizing the growth and spread of Legionella in at-risk water systems. This can include regular cleaning and disinfection of cooling towers and other industrial water systems, maintaining hot water tanks at temperatures above 60 degrees C, that's 140 Fahrenheit, which inhibits Legionella growth, and using disinfectants like chlorine dioxide or copper-silver ionization to treat water.
Moving on to typhoid fever, which our sources tell us is caused by a specific type of salmonella called Salmonella enterica typhi. This also sounds like a serious waterborne illness, especially prevalent in areas with inadequate sanitation.
Yes, typhoid fever is a systemic infection caused specifically by Salmonella typhi. It's transmitted through the ingestion of water or food that has been contaminated with the feces of someone infected with the bacteria. This is why it remains a significant public health concern in areas where sanitation and hygiene are poor. Once someone ingests the bacteria, they travel to the small intestine, can cross the intestinal lining, and then spread through the lymphatic system and bloodstream to various organs, leading to a widespread infection throughout the body.
What are the typical symptoms of typhoid fever and how is it treated?
The symptoms of typhoid fever usually develop gradually, starting with a low-grade fever and a general feeling of being unwell, which then progresses to a high fever, headache, abdominal pain, and potentially constipation or diarrhea. If left untreated, it can lead to severe complications. Treatment involves antibiotics that are effective against Salmonella typhi, such as ciprofloxacin or ceftriaxone, along with supportive care like ensuring adequate hydration. It's also important to note that some people can become asymptomatic carriers after recovering from typhoid fever, meaning they no longer show symptoms but still carry the bacteria, often in their gallbladder, and can continue to shed it in their feces, posing a risk of transmission to others.
Oh, wow. Carriers.
Yeah. Prevention, as with many waterborne illnesses, relies heavily on improved sanitation and effective water treatment.
Finally, in our discussion of waterborne diseases, we have norovirus illness. This is a virus, and our sources indicate it's a very common cause of gastrointestinal illness, often associated with contaminated water or food.
That's correct. Norovirus is a single-stranded RNA virus and a major cause of what we often refer to as stomach flu. It targets the cells lining the small intestine. The main symptoms are typically vomiting, diarrhea, nausea, and abdominal cramps, which usually last for a relatively short period, often between 12 and 72 hours.
Our sources mention that norovirus has a very low infectious dose. It only takes a tiny number of viral particles to make someone sick. That sounds incredibly contagious.
It is highly contagious, and that low infectious dose is a key reason why it spreads so easily. Because it takes so few viral particles to cause infection, even a small amount of contamination can lead to widespread outbreaks. Transmission primarily occurs through the fecal-oral route, meaning the virus is shed in the feces or vomit of an infected person, and can then be inadvertently ingested through contaminated water, food, or even by touching contaminated surfaces and then your mouth.
Ugh.
Common waterborne sources of norovirus outbreaks include contaminated well water and recreational waters.
How is norovirus illness usually diagnosed, and what's the approach to treatment?
Diagnosis is often based on the characteristic symptoms, particularly the prominent vomiting. However, especially in outbreak situations or to confirm the diagnosis, viral RNA or antigens can be detected in stool or vomit samples using laboratory tests like PCR. Unfortunately, there's no specific antiviral medication for norovirus. Treatment focuses on supportive care, mainly ensuring that the affected person stays well hydrated to replace the fluids lost through vomiting and diarrhea.
So a very unpleasant and easily spread virus. Okay, we've explored how water can act as a carrier of disease and some of the key illnesses it can transmit. Let's now turn our attention to food. Our sources emphasize that foods are rarely sterile and can harbor both spoilage organisms and actual disease-causing pathogens. This really highlights the critical importance of proper food storage and preparation.
Absolutely. Unlike the rigorous treatment processes that drinking water undergoes in developed countries, our food supply chain, from farm to table, presents numerous opportunities for microbial contamination. Therefore, understanding the factors that lead to food spoilage and the methods we can use to preserve food are crucial for preventing illness and ensuring food safety.
Let's begin with food spoilage. It's defined as any changes that make food unacceptable for consumption. And our sources categorize foods based on their perishability, linking this to the concept of water activity. Can you explain what water activity is and how it relates to how quickly food spoils?
Sure. Water activity, often abbreviated as SW, is essentially a measure of how much unbound water is available in a food for microorganisms to grow. Perishable foods like fresh meats, poultry, dairy, and produce have a high water activity, making them very attractive to a wide variety of bacteria and fungi that cause spoilage. These foods have a short shelf life and typically require refrigeration or other preservation methods to remain safe and palatable. Semi-perishable foods, such as some fruits and vegetables or cured meats, have an intermediate water activity and a limited shelf life. Non-perishable foods, like dried grains, and properly canned goods, have a very low water activity, making it difficult for most microbes to grow, which gives them a long shelf life. The specific types of microorganisms that cause spoilage in fresh foods depend on factors like the food's nutrient content, its pH, and the temperature at which it's stored.
That makes sense. The more available water, the more hospitable the environment for microbial growth. Now, what are the main ways we preserve food to prevent the spoilage and, more importantly, to keep it safe to eat and prevent foodborne illnesses?
Food preservation methods essentially work by either killing any microorganisms present in the food, or by creating conditions that inhibit their growth. Altering the temperature is a primary strategy, including refrigeration to slow down microbial metabolism significantly, freezing to essentially stop it, heating like in cooking, canning, or pasteurization to kill or significantly reduce the number of microbes. Pasteurization is particularly important for heat-sensitive liquids like milk and juice.
Hmm.
Reducing the availability of water is another key approach, achieved through methods like drying or by adding solutes, such as salt or sugar, which draw water out of microbial cells through osmosis. Irradiation, specifically using electron beam or E-beam technology, can also be used to sterilize certain foods. Additionally, antimicrobial chemicals can be added as preservatives, and fermentation, which involves the controlled growth of beneficial microorganisms, can produce natural preservatives like lactic acid or alcohol, enhancing both the safety and the flavor of foods like yogurt, cheese, and sauerkraut.
So there's quite a variety of techniques we can use to extend the shelf life and safety of our food. Now let's shift our focus to actual foodborne diseases. Our sources categorize these into two main types: food poisoning or intoxication, which results from ingesting preformed toxins, and food infection, which occurs when we ingest live pathogens that then grow and multiply in our bodies. Interestingly, the information indicates that food infections are more common in the US. Why might that be?
That's a good question. It likely reflects a combination of factors, including the types of foods we commonly consume, our food handling practices, and the prevalence of certain pathogens in our food supply. Food infections often have a longer incubation period because the bacteria or viruses need time to multiply in the body before causing symptoms, so they might be statistically more frequently identified in surveillance data.
And our sources list the big 8 microorganisms that are responsible for the vast majority of foodborne illnesses. It's a notable list: Salmonella, Clostridium perfringens, Campylobacter jejuni, Staphylococcus aureus, Listeria monocytogenes, Escherichia coli, norovirus, and Toxoplasma. We'll be discussing many of these in more detail. But first, how do scientists actually go about taking samples of solid foods to detect these microbes, especially when they're investigating an outbreak? The paddle blender, or stomacher, gets some mention. What's the purpose of this tool?
When you need to analyze a solid food sample for the presence of microorganisms, you first need to get those microbes off the food and into a liquid medium where they can be easily studied. The stomacher is a device that helps with this process. You place a small sample of the food into a sterile bag along with a sterile liquid. The stomacher then uses paddles to vigorously crush and mix the food sample within the bag.
Ah, like a mini blender.
Kind of, yeah. This action effectively dislodges any bacteria or other microbes that might be present on or in the food, releasing them into the liquid. This liquid can then be used for further microbiological analysis, such as culturing the bacteria on growth media or using molecular tests to identify specific pathogens.
So it's like giving the food a really thorough shake to get any hidden microbes out and into a state where they can be detected. And the information emphasizes that the gold standard for confirming that a particular food caused an outbreak is identifying the exact same strain of pathogen in both the sick individuals and the suspected food product. How do scientists achieve that level of precision?
You're right, that's the most definitive way to link a food item to an illness outbreak. Scientists use advanced molecular techniques to generate a unique fingerprint of the DNA of the bacteria or virus isolated from both the patients and the food. Techniques like pulsed field gel electrophoresis, PFGE, or increasingly whole genome sequencing can provide very detailed genetic information. If the genetic fingerprint of the pathogen from the sick people perfectly matches the fingerprint of the pathogen found in the suspected food, it provides very strong evidence that the food was indeed the source of the outbreak. It's like matching DNA at a crime scene.
Now, our sources also touch on the field of foodborne disease epidemiology, how these outbreaks occur, how they are tracked and investigated, and the important roles of food epidemiologists and surveillance networks like FoodNet and PulseNet. It sounds like a really crucial system for protecting public health.
It is absolutely vital. Foodborne disease outbreaks can happen in a wide variety of settings: restaurants, catered events, even in our own homes. The way our modern food system is structured, with centralized processing and distribution, means that contamination at a single point can potentially lead to illnesses across many different states or even countries.
Right, that makes sense.
Food epidemiologists are like disease detectives. When an outbreak is suspected, they work to identify the source of contamination, the specific food involved, and the factors that might have contributed to the problem. Surveillance networks like FoodNet, which actively tracks foodborne illnesses in certain areas of the U.S., and PulseNet, which maintains a national database of the DNA fingerprints of foodborne bacteria, are essential tools in this process. They help to detect outbreaks early, link seemingly unrelated cases to a common source, and enable public health officials to take timely action to prevent further illnesses, such as issuing recalls or implementing control measures in food production facilities.
It's reassuring to know that there are these sophisticated systems in place to monitor and respond to these issues. Okay, let's now delve into the specifics of food poisoning, starting with staphylococcal food poisoning. Our sources tell us this is caused by heat-stable enterotoxins produced by Staphylococcus aureus. That heat-stable part sounds significant.
It is a key characteristic. Staphylococcus aureus is a common gram-positive bacterium that can be found on our skin and in our noses. If food becomes contaminated with S. aureus and is then left at room temperature for a period of time, the bacteria can multiply and produce toxins called enterotoxins, with staphylococcal enterotoxin A, SEA, being the most common. What makes these toxins particularly problematic is that they are heat-stable, meaning they can withstand the temperatures typically used for cooking.
Ah, so reheating doesn't help.
Exactly. Even if cooking kills the Staphylococcus bacteria, if toxins were already produced in the food, those toxins can still be present and cause illness when the food is consumed.
And how quickly do the symptoms of staph food poisoning usually appear, and what kind of symptoms are we talking about?
The onset of symptoms is typically quite rapid, usually within 30 minutes to 6 hours after eating the contaminated food. The symptoms are primarily gastrointestinal and can include nausea, vomiting, abdominal cramps, and diarrhea. Dehydration can also occur as a result of the fluid loss.
How does the food usually get contaminated with Staphylococcus aureus in the first place? Our sources mention food preparers.
Yes, a very common source of contamination is food handlers who carry S. aureus on their skin or in their nasal passages. If they don't follow proper hygiene practices, such as thorough hand washing, they can transfer the bacteria to food, especially foods that are handled after cooking and don't undergo any further heating. Foods that are particularly risky include custard-filled baked goods, poultry, eggs, and mayonnaise-based salads, essentially foods that are often prepared with a lot of handling and are left at room temperature.
And what's the treatment for staph food poisoning? Since it's the preformed toxin causing the issue, I would guess that antibiotics aren't very effective.
You're absolutely right. Because the illness is caused by the ingestion of the preformed toxin rather than an active bacterial infection in the body, antibiotics are generally not helpful. Treatment focuses on supportive care, primarily resting and replenishing lost fluids and electrolytes to prevent dehydration. The illness is usually relatively short-lived, typically resolving within 24 to 48 hours. Prevention is key and relies on strict hygiene and sanitation practices by food handlers, as well as ensuring that susceptible foods are properly refrigerated to prevent bacterial growth and toxin production.
Okay, that makes sense. Now let's move on to clostridial food poisoning, which our sources indicate is caused by toxins produced by endospore-forming anaerobic bacteria, specifically Clostridium perfringens and Clostridium botulinum. Let's start with C. perfringens.
Clostridium perfringens is a bacterium that is commonly found in soil and in the intestinal tracts of animals and humans. Unlike Staphylococcus aureus, food poisoning from C. perfringens typically requires ingesting a large number of the bacterial cells. This often happens when cooked foods, particularly high-protein items like meats and poultry, are left at room temperature for an extended period. This allows surviving C. perfringens spores to germinate and the bacteria to multiply to levels high enough to cause illness.
So temperature control is key here.
Absolutely. The illness itself is caused by a toxin called perfringens enterotoxin, which is produced in the small intestine during the process of sporulation, the formation of endospores by the bacteria.
What are the common symptoms of C. perfringens food poisoning, and how is it typically diagnosed and prevented?
The symptoms usually include nausea, diarrhea, and abdominal cramps, and they typically begin 6 to 24 hours after consuming the contaminated food. Vomiting and fever are less common with this type of food poisoning. Diagnosis can sometimes be suspected based on the symptoms and the relatively late onset, but it can be confirmed by isolating large numbers of C. perfringens from the patient's stool or by detecting the enterotoxin in fecal samples. Prevention is crucial and relies on proper food handling practices. Cooking foods to a safe internal temperature and, very importantly, rapidly cooling and properly refrigerating any cooked foods that are not going to be served immediately to prevent the germination and growth of C. perfringens. Keeping hot foods hot and cold foods cold is the key.
Now, the other significant player in clostridial food poisoning is Clostridium botulinum, which causes the very serious condition of botulism. Our sources emphasize that this is due to a potent neurotoxin. That sounds quite different from the toxins we've discussed so far.
It is significantly different and potentially much more dangerous. Clostridium botulinum is another anaerobic endospore-forming bacterium, commonly found in soil and water. The botulinum toxin it produces is one of the most potent neurotoxins known. Foodborne botulism typically results from consuming improperly processed, low-acid canned foods, such as home-canned vegetables like corn or beans, where the anaerobic conditions inside the botulina can allow C. botulinum spores to germinate and produce the deadly toxin.
Home canning, right.
Yes. Even a tiny amount of this toxin can cause severe illness by blocking nerve function, leading to flaccid paralysis, a muscle weakness that often starts in the face and then progresses down the body, eventually affecting the muscles used for breathing and potentially leading to respiratory failure and death.
That sounds incredibly serious. Our sources also mention infant botulism and wound botulism. Are those caused by the same mechanism as foodborne botulism?
While all types of botulism are caused by the botulinum toxin produced by Clostridium botulinum, the way the toxin gets into the body differs. Foodborne botulism, as we discussed, involves ingesting food containing the preformed toxin. Infant botulism occurs when infants, typically under 1 year old, ingest C. botulinum spores, which then germinate and produce the toxin directly in their immature intestines. Honey is a well-known risk factor for infant botulism because it can sometimes contain C. botulinum spores.
Ah, the honey warning for babies.
Exactly. Wound botulism is less common and happens when C. botulinum endospores contaminate a wound and produce the toxin there, which then enters the bloodstream.
How is botulism typically diagnosed and treated? And what are the critical steps for preventing it?
Diagnosis can be challenging in the early stages, as the symptoms can sometimes mimic other neurological disorders. It often involves detecting the botulinum toxin in the patient's serum, stool, or in the suspected food. Identifying the C. botulinum bacteria in the stool or wound can also be helpful. Treatment involves administering botulinum antitoxin, which can neutralize the circulating toxin but cannot reverse any paralysis that has already occurred. Supportive care, including mechanical ventilation if breathing is affected, is also absolutely crucial. Prevention of foodborne botulism relies heavily on using proper techniques for home canning of low-acid foods, ensuring that they are processed at high enough temperatures and pressures to kill C. botulinum spores. For infants, avoiding giving them honey is a key preventative measure against infant botulism. Proper cleaning and care of wounds can help prevent wound botulism.
So, botulism is a very serious and potentially life-threatening condition that requires prompt medical attention.
Okay, we've thoroughly covered food poisoning. Let's now turn our attention to food infection, where we're actually ingesting live pathogens that then colonize and multiply within our bodies, leading to illness. We'll start with salmonella and salmonellosis, which our sources tell us is a very common gastrointestinal disease.
Yes, salmonellosis is a widespread bacterial foodborne illness caused by ingesting salmonella bacteria, which are gram-negative and commonly found in the intestinal tracts of many animals, including poultry, livestock, and reptiles. They could also contaminate the environment through animal feces. There are many different types, or serovars, of Salmonella enterica, with serovars like typhimurium and enteritis being frequently implicated in foodborne outbreaks. Infection occurs when we eat food that has been contaminated with Salmonella, typically through fecal contamination, either originating from animal sources during food production or from food handlers with poor hygiene. Once ingested, the Salmonella bacteria travel to the small intestine, where they attach to and invade the cells lining the intestine. They can also be taken up by immune cells. The bacteria then multiply and release various substances that cause inflammation, leading to enterocolitis inflammation of the small and large intestines and the characteristic symptoms of salmonellosis.
And what are those typical symptoms of salmonellosis, and when do they usually start after someone eats contaminated food?
The symptoms usually include diarrhea, which can sometimes be bloody vomiting, abdominal cramps, and fever. They typically begin anywhere from 12 to 72 hours after consuming the contaminated food.
How is salmonellosis typically diagnosed and treated, and what are the most important things we can do to prevent it?
Diagnosis usually involves culturing Salmonella bacteria from a stool sample in the laboratory using specialized growth media. For most cases of uncomplicated salmonellosis, treatment is primarily supportive, focusing on rest and drinking plenty of fluids to prevent dehydration. Antibiotics are generally not recommended for mild cases, as they don't usually shorten the duration of the illness, and in some cases might even prolong the time the bacteria are shed in the stool.
Interesting.
Prevention is really key and relies on practicing proper food handling and cooking techniques, such as cooking poultry, meat, and eggs to safe internal temperatures, preventing cross-contamination between raw and cooked foods, for example, by using separate cutting boards, and always washing your hands thoroughly before preparing or eating food.
Now, let's talk about another very well-known bacterium, Escherichia coli. Our sources point out that while most strains of E. coli are harmless and live in our intestines, some are pathogenic and can cause foodborne and sometimes waterborne illnesses. We have Shiga toxin-producing E. coli, or STEC, also known as enterohemorrhagic E. coli, or EAC, getting a specific mention. The second name, enterohemorrhagic, really sounds alarming.
It is a particularly concerning group of E. coli. STEC, and especially the serotype O157H7, are notorious because they produce a potent toxin called verotoxin, which is very similar to the Shiga toxin produced by Shigella bacteria. This toxin can cause significant damage to the lining of the intestines, often leading to bloody diarrhea. In some cases, particularly in young children, the elderly, and those with weakened immune systems, STEC infection can also lead to a very serious and potentially life-threatening complication called hemolytic uremic syndrome, or HUS, which can cause kidney failure.
Kidney failure. Wow.
Yeah. That's why the hemorrhagic part of the name is so significant. It refers to the bloody diarrhea, and the risk of kidney complications makes it a serious concern. It's important to understand that STEC and EAC are essentially different names for the same group of dangerous E. coli.
Our sources frequently link E. coli O157.H7 to undercooked ground beef. Why is that such a common source of infection?
Cattle can carry E. coli O157.H7 in their intestines without showing any signs of illness. During the slaughtering process, there's a risk of fecal contamination of the meat. When beef is ground, any bacteria that were present on the surface of the whole cut of meat get mixed throughout the entire ground product. If this ground beef is not cooked thoroughly to an internal temperature of 160 degrees AFRI, that's 71 degrees C, these E. coli bacteria can survive and cause infection when the meat is eaten.
That mixing process is key.
It really is. Other sources of STEC outbreaks can include raw milk, contaminated fresh produce like spinach and lettuce, often contaminated through irrigation water or animal runoff, and even contaminated water sources.
How is a STEC infection typically diagnosed and treated? And are antibiotics a helpful treatment in these cases?
Diagnosis usually involves testing a stool sample to culture E. coli, and then specifically looking for the presence of the Shiga toxin, or identifying the O157.H7 serotype. Treatment for STEC infection is primarily supportive, focusing on ensuring adequate hydration and closely monitoring for and managing any potential complications, such as hemolytic uremic syndrome. Importantly, antibiotics are generally not recommended for STEC infections and may even increase the risk of developing HUS by potentially triggering the release of more toxin.
So antibiotics could make it worse.
Potentially, yes. Prevention is critical and centers on thoroughly cooking ground beef and other meats, washing raw fruits and vegetables very well, avoiding unpasteurized milk and dairy products, and practicing good hygiene, especially after using the restroom and before preparing food.
Our sources also briefly mentioned some other types of pathogenic E. coli. Enterotoxigenic E. coli, ETEC, enteropathogenic E. coli, EPEC, and enteroinvasive E. coli, EAC. Can you give us a quick overview of how these differ from STEC?
Certainly. ETEC is a major cause of traveler's diarrhea and produces toxins that lead to watery diarrhea. EPEC primarily causes diarrhea in infants and young children through a different mechanism involving the bacteria attaching very closely to the cells of the intestinal lining and disrupting their normal function. EIC is similar in its effects to Shigella. It invades the cells of the colon lining, causing watery diarrhea that can sometimes be bloody, along with fever and abdominal cramps. Each of these groups of pathogenic E. coli has different specific mechanisms of causing disease and different virulence factors compared to STEC.
Okay, so a diverse and sometimes dangerous family of bacteria with different ways of making us sick. Now, our sources state that Campylobacter infection is now the most frequently reported bacterial cause of foodborne illness in the United States. It's a significant piece of information.
It is a very common bacterial infection. Campylobacter jejuni and C. fetus are the species most often linked to human illness. A major source of C. jejuni is poultry, as it is often found as part of the normal bacterial flora in their intestines. Transmission to humans typically occurs through consuming undercooked poultry or pork, raw or undercooked shellfish, or drinking water that has been contaminated with animal feces. Even handling raw poultry and not washing your hands thoroughly afterwards can lead to infection.
What are the typical symptoms of a Campylobacter infection?
Ingestion of Campylobacter leads to its multiplication in the small intestine, causing inflammation. The symptoms typically include fever, headache, abdominal pain, and diarrhea, which is often bloody. The illness usually lasts for about a week, and in some cases, it can be followed by more serious complications like Guillain-Barré syndrome.
Guillain-Barré, that's a neurological issue, isn't it?
It is, yes. It's an autoimmune disorder that can cause muscle weakness and sometimes paralysis. It's a rare but known complication following Campylobacter infection.
How is Campylobacter infection usually diagnosed and treated, and what can we do to prevent it?
Diagnosis involves isolating Campylobacter bacteria from a stool sample using specialized selective media in the lab. In most cases, the illness is self-limiting and only requires supportive care, such as drinking plenty of fluids and getting rest. However, in more severe cases or in individuals with weakened immune systems, antibiotics like azithromycin or ciprofloxacin may be prescribed. Prevention strategies are very similar to those for other foodborne bacteria, emphasizing the importance of thoroughly cooking poultry and other meats to the correct internal temperature, preventing cross-contamination between raw poultry and other foods, and ensuring the safety of drinking water. Good hand hygiene is also crucial, especially after handling raw poultry.
Let's move on to Listeria monocytogenes and the illness it causes, listeriosis. Our sources highlight that while it might cause relatively mild symptoms in healthy individuals, it can be very serious, even life-threatening in certain susceptible populations.
That's a very important point. Listeria monocytogenes is a bacterium that has the somewhat unusual ability to grow even at refrigerator temperatures, which makes it a particular concern for certain types of ready-to-eat foods.
Grows in the cold. That's tricky.
It is. It's found widely in the environment, including soil and water, and can contaminate a variety of foods, particularly ready-to-eat deli meats and hot dogs, soft cheeses, unpasteurized dairy products, and inadequately pasteurized milk. In healthy adults, infection with Listeria might cause only mild flu-like symptoms or even be asymptomatic. However, in pregnant women, newborns, the elderly, and people with weakened immune systems, listeriosis can lead to serious invasive infections, including bacteremia, infection of the bloodstream, and meningitis, inflammation of the membranes surrounding the brain and spinal cord, which can have a high mortality rate. Listeria infection during pregnancy can also lead to miscarriage, stillbirth, or severe illness in the newborn.
Our sources also mention that Listeria has some clever ways of evading the body's immune defenses. That sounds quite sophisticated for a bacterium.
It is. Listeria has evolved several mechanisms to survive and spread within the host.
Right.
It can invade phagocytic immune cells, but then has the ability to escape from the phagosome, the compartment within the cell, where pathogens are typically destroyed using a protein called Listeriolicin O.
It breaks out.
Essentially, yes. Once it's in the cytoplasm of the host cell, it can multiply and then use another protein called ACD to propel itself and spread directly from one host cell to another without ever being exposed to the extracellular immune defenses. This cell-to-cell spread is a key factor in its ability to cause systemic infections.
How is listeriosis typically diagnosed and treated? And what are the main efforts to prevent it?
Diagnosis of invasive listeriosis usually involves culturing Listeria bacteria from blood or cerebrospinal fluid. Treatment typically involves antibiotics, such as penicillin or ampicillin, often in combination with an aminoglycoside like gentamicin. Prevention is critical and includes food recalls of contaminated products and ongoing efforts to minimize contamination in food processing environments. For individuals who are at higher risk of severe listeriosis, public health recommendations include avoiding certain high-risk foods, such as unpasteurized dairy products and ready-to-eat meats that haven't been thoroughly heated. Pregnant women are often advised to avoid soft cheeses and deli meats unless they are heated until steaming hot just before consumption.
We've covered quite a few of the key bacterial food infections. Our sources also provide a brief overview of some other foodborne infectious diseases, including Yersinia enterocolitica, Bacillus cereus, Shigella, and various Vibrio species, as well as viral and protist pathogens. It seems like there's a very wide range of potential microbial threats lurking in our food.
It is a diverse range indeed. Yersinia enterocolitica, which can be found in contaminated meat and dairy products, can cause gastrointestinal illness and sometimes symptoms that mimic appendicitis. Bacillus cereus is interesting because it's an endospore-forming bacterium that can produce different types of toxins depending on the conditions, leading to either vomiting or diarrheal-type illness, often associated with cooked rice and other starchy foods left at room temperature.
Ah, the fried rice syndrome.
That's the one. Shigella bacteria cause shigellosis, which is often spread through the fecal-oral route, but can also be transmitted through contaminated food and water, leading to dysentery with bloody diarrhea. Various Vibrio species, other than Vibrio cholerae, can cause food poisoning, primarily from eating raw or undercooked shellfish. Viruses like norovirus, which we also discuss as a waterborne pathogen, rotavirus, astrovirus, and hepatitis A, often transmitted through contaminated shellfish or raw produce, are also significant causes of foodborne illness.
And protists too.
Yes. Then we have protists, which are single-celled eukaryotic organisms such as Giardia intestinalis, Cryptosporidium parvum, Cephalospora chitinensis, and Toxoplasma gondii. These can contaminate food and water through various routes, including contaminated water or contact with cat feces in the case of Toxoplasma.
And finally, in a category all its own, our sources mention prions, such as those responsible for variant Creutzfeldt-Jakob disease, VCJD, linked to beef contaminated with bovine spongiform encephalopathy, BSE, or mad cow disease. It's noted that prions are unique because they are neither cellular organisms nor viruses.
That's right. Prions are misfolded proteins that have the ability to induce normal proteins to also misfold, leading to a chain reaction of protein misfolding and the development of fatal neurodegenerative diseases. They're distinct from bacteria, viruses, and parasites because they don't contain any genetic material. Their unique nature makes them particularly challenging to detect and inactivate.
Well, that's a truly comprehensive and, at times, sobering overview of the microbial threats that can be present in our water and food. It's a lot of information to absorb, but it's incredibly important to have this understanding.
It is, and the key takeaway is the remarkable diversity of these microbial agents and the many different ways they can be transmitted to us through our daily consumption of water and food. Understanding these transmission pathways is fundamental to implementing effective prevention strategies, both at the level of public health and in our own individual behaviors.
As we've discussed, there's a clear distinction to be made between illnesses transmitted through water and those transmitted through food, and within foodborne illnesses, between food poisoning caused by preformed toxins and food infection resulting from live pathogens multiplying in the host. Recognizing these fundamental differences helps us understand how these illnesses manifest and how they are best addressed.
That's exactly. Robust public health measures, such as ensuring clean water supplies, implementing effective sanitation systems, and enforcing stringent food safety regulations, are absolutely critical. But equally important is individual awareness and the consistent practice of safe food handling and good personal hygiene in our own homes. These multiple layers of defense are what ultimately protect us from these microscopic threats.
So, thinking about everything we've discussed, particularly the interconnectedness of our global food and water systems and that intriguing example of reverse zoonosis in Antarctica, it really prompts a larger question. Considering ongoing global changes such as climate change, increased international travel and trade, and evolving practices in agriculture and food production, how might these factors influence the emergence and spread of these waterborne and foodborne diseases in the years to come? It's definitely something to consider.
That is a profoundly important question to contemplate. The interconnectedness you mentioned, combined with significant environmental and societal shifts, could indeed create new pathways for these pathogens to emerge, evolve, and spread in potentially unexpected ways. Continuous scientific research, robust surveillance systems, and proactive public health strategies will be absolutely essential to address these future challenges.
Well, I think it's clear that this deep dive has thoroughly explored the key aspects of the material you provided on waterborne and foodborne bacterial and viral diseases. It's been truly eye-opening to understand the complexities involved in ensuring the safety of our water and food supply and the many ways these microscopic organisms can impact our health.